Okay, so here are two good examples of how structure relates directly to function in the nervous system. The first is actually something called an en passant synapse, and there is a description and an image actually on our course website. But I want to sort of describe it in words too. So, an en passant synapse. En passant is actually maybe a term that's familiar to those of us who play chess. It's when you take a pawn and you move past it. When it's its first move, you can move either one or two spaces. If you go past it, you can actually take the other guy's pawn, the other person's pawn, in passing or en passant. And how it relates to the nervous system is that in some neurons, actually, there are these swellings along the axon. So, the axon hillock, right? Here we go, right? It leaves, the cell body here. There's a swelling at the beginning, studded with voltage-gated sodium and potassium channels to help initiate, the action potential. If you reach the threshold of the axon hillock, you're going to get an action potential that'll run the length of the axon. But there's going to be this sort of initial swelling, then another swelling, then another swelling. And at all of these kinds of swellings along the axon, you find vesicles. You find the docking complex proteins. You find voltage-gated calcium channels. You find all the machinery that's typically associated with, the release of neurotransmitter at the axon terminal. But in this case, it's located multiple places along the swellings on a single axon. So this means that when you depolarize at the axon hillock to -55 and you open all those voltage-gated sodium and potassium channels, and the sodium current, the depolarizing wave starts working its way down, you're going to depolarize to the threshold for calcium channels at each of these swellings, right? You're going to get calcium entry, you're going to get vesicle fusion, you're going to get exocytosis and release of neurotransmitter. In passing, as the depolarizing wave works its way down towards the physical axon terminal, these are like functionally axon terminals all the way along at each of these swellings. So what happens is you get stimulation, and you get, it's like a soaker hose, you get neurotransmitter, and there's lots of synaptic connections made all the way along this axonal structure with all the various swellings and the release machinery located at each one. So these are called en passant synapses that are formed, and there's a particularly good example of this architecture in the locus coeruleus, which is in the brainstem. There are only about 5,000 noradrenergic neurons in the left and in the right, and they project pretty widely around the cortex and around the brain, but they subserve a pretty critical sort of function, right? I apologize, but there's a sudden startle synapse, something, or startle stimulus, something that wasn't expected, something novel or unusual or unexpected. You get powerful activation of the locus coeruleus and release of norepinephrine all the way along these swellings, all these en passant synapses, and at the actual axon terminals at the very end. It exaggerates and amplifies the influence of these 5,000 neurons. They can generate a pretty powerful response because just the activation of a single one, releases norepinephrine at a multitude of synapses at all these en passant locations along the way. The other good example of structure-function relationships is how neuroscientists were really interested when they put in an electrode, to stimulate, one neuron. Let's see here, right? The wire, the axon of one, and then they recorded postsynaptically from the next. They would record, if it's an excitatory neurotransmitter, acting on an ionotropic receptor that, let's say, opens up a calcium or a sodium, channel, you'd see an excitatory postsynaptic potential in the next cell. And, they would try to get it to the barest minimum. And what they found was that the EPSP response to different levels of stimulation in the presynaptic element, it wasn't kind of a continuous response. It didn't go up, just a little bit. It went up by what we refer to as quantal units. It would the EPSP would jump, and then it would jump, and then it would jump as you increase the stimulation presynaptically. And you couldn't get it down to just above zero. It would be some basic quantal unit of excitatory postsynaptic potential response. And it's that behavior, that the excitatory response or the inhibitory response, it turns out, of an excitatory or inhibitory neurotransmitter is due to the fact that you can't just release a single molecule of, let's say, glutamate or GABA, right? You release glutamate or GABA in vesicle size amounts, right? The individual vesicles contain 100 molecules, let's say, of glutamate. That's going to produce a particular, quantal response, like a certain unit of response in terms of the excitatory postsynaptic potential. If you release 2 vesicles, you double it, right? If you release 3 vesicles, you triple it. But it's not a continuous EPSP function. It jumps in these quantal units. So there is a quantal nature to the responsiveness of neurons to the release of neurotransmitter because of how neurotransmitter is stored structurally in vesicles. You can never release less than 1 vesicle.